Good morning. So Mike, I think, gave a great introduction to enzyme replacement therapy. I'm here to introduce you to Valerion Therapeutics, formerly Forest 3 Bioscience, which I think many of you have heard of. And as Mike said, Dustin Armstrong really wanted to be here, but I made him stay in the lab with Alan Beggs, redoing the mouse study that Mike just actually presented to you. So he's busy injecting mice and watching them run around the cages, we hope. Anyway, the reason we changed our name to Valerion is because Valerion comes from the Italian verb valere, which stands for to be healthy and strong. And we're working in the neuromuscular disease field, and that's what we want for our patients. So, very quickly, small biotech company. We are based in Concord, Massachusetts. The Wards actually visited us a few weeks ago. Any of you are welcome to visit our offices. They're tiny little offices in the basement of the building, but we are real. We have a website. Anyway, we were founded in 2007 by Dustin, and over that time we've been awarded 2 MDA grants totaling $1.5 million to study specifically myotubular myopathy. We also last year received $20 million from a family fund, which I'll talk briefly about, to accelerate development of a drug for MTM. The family fund is the Pritzker family out of Chicago, and they recently developed a health fund back in 2011. They have invested in 4 companies. We are by far the largest investment, and they are absolutely dedicated to getting a treatment for MTM with their money. We have the funding, we will continue to get the funding, and they're very supportive of us. So we need to know that too. Mike actually talked about this, so I'm just going to tell you, we have an antibody that penetrates muscle cells preferentially. We can link myotubularian to this antibody, creating a fusion protein, and deliver myotubularian directly into muscles. Why MTM? First of all, as Mike said, it's a disease that's classic to, it's amenable to protein replacement therapy. We know which protein is missing, number 1. Number 2, we can target that protein to where it needs to be, to the muscle. We know we can do that, we've shown it in the mice. And number 3, as Mike said, and Anna said, we have excellent mouse models for both gene therapy and protein therapy that show a muscle pathology in a clinical course that is similar to the human disease, which means hopefully the data we have from the mice will be translatable to human studies. So where are we? Right now we're designing our fusion protein. It is in moderately early stage. It's still a research project. We are using several different types of cell lines to see which cell line actually produces a protein that we can make it, commercialize it, make it not inexpensively, but for the cost of goods is such that you can afford it. And that will probably take us about 6 to 9 months to finish that process. At that point, we're hoping we'll have a stable manufacturing process. That won't mean we'll have clinical material, but we'll have the blueprint for us to be able to start making clinical material. And we've actually made some early material that Dustin's testing in the animals right now with one of our cell lines. I'm not sure it's the perfect drug, but it might be good enough. So we may choose to move forward with that drug and take that into clinical studies as well. Once we have that product, we'll begin safety testing, which Casey are already involved in. And we'll do that in animal species to prepare for human studies, and then we'll go into the clinic. And as Anna said, it's really hard to predict how long it will take, so I'm not going to try to do that. But the key will be getting that fusion protein designed, getting it into a cell line that can produce it efficiently, and then we'll know from there. We should know that later on this year. We should have ahead. We're also supporting a large amount, frankly, of MTM research initiatives. The CMDIR genetic testing protocol. Sabine is here. Where is she? Sabine is right there. She has 5 genetic testing kits with her. She has patient-informed consents with her. I encourage you very, very strongly to get tested. We need to understand the mutations. We need to know what you have. We need to confirm that. It helps you and it helps us. Also, the phone-based event study, again, sponsored by CMDIR and Jim Dowling, Jim's natural history study, Jim's biomarker study, and lastly, a functional study that we are co-funding and co-sponsoring with Genathon and Institut Immunologie. We're going to be running that study both in North America at the NIH and with Jim, and also in Europe with Laurent Sauvé and Anna Buchabello. And I'm going to let Laurent come up and talk a little bit about this functional study. But I want to give one slide before he does, but I want him to get his stuff together. This is not a natural history study. This is a longitudinal functional study. And the purpose of this study is to examine and quantify functional outcomes in untreated patients over time. And then, when we design our clinical trials, we use those same functional outcomes over that period of time to look for improvements. And the reason, or part of the reason that we need to do this is, number one, to practice with those functional measurements and see if we're able to do them consistently in clinical study. Hal actually is going to speak about this a bit tomorrow, so you're going to get more detail about the study tomorrow. But what we want this process to do is help us to educate FDA, EMA, Health Canada about MTM. You're a rare disease. They're not going to have an expert at the agencies. It's our job to educate them about the disease. And actually, it's also our job to show them how improving strength and muscle function in the patients, and you, and your family members, will benefit patient lives, because that will be their question. In order to get a drug approved, we have to show that we change a patient's life, and we show a benefit. So the purpose of this study is to do that. It is really important that we do this study. Anyway, Laurent.